Hello medicos, let us start from the embryology of eye. So the eye originates from, or the tissue of eye originates from surface ectoderm, neuroectoderm, then we have mesoderm, and the fourth is neural crest cells. In this lecture, we are going to see how all these four layers give rise to eyeball plus orbit plus extraocular muscles. Okay, so now let us start with the surface ectoderm. So the first topic, let us put it as surface ectoderm. Before moving towards the list of the structures which came from surface ectoderm, you need to know some things like, see, this is neuroectoderm. Considering this as a neuroectoderm, and in red color it is surface ectoderm. So SE for surface ectoderm and NE for neuroectoderm. From this surface ectoderm, there is a formation of or protrusion of a structure which is called as optic stalk. And then we have a surface ectoderm. This optic stalks continues to grow towards the surface ectoderm. So now we have an optic stalk and a surface ectoderm which proliferates and forms a pit-like structure. So this is a surface ectoderm, and this surface ectoderm forms a pit. And this is called as lens pit. Okay. So what do we have? We have a neuroectoderm from which we have a bulging structure which is called as optic stalk. This optic stalk tends to grow towards the surface ectoderm, and after the proliferation of this surface ectoderm, we have a lens pit. Now what happens? So we have an optic stalk over here. This near this pit, this surface ectoderm turns to be proliferating, which forms the lens placode. What you call it as? Lens placode. Okay. So the tissues near the lens pit tends to proliferate to form the lens placode. Now what happens? After some time, this lens placode pinch off from this layer of surface ectoderm and dive inside this neuroectoderm. So what do we get here? This is neuroectoderm, and meanwhile this neuroectoderm form a cup-like structure. Okay, so we have lens vesicle or lens proper. Now this remaining surface ectoderm give rise to corneal epithelium and the conjunctival epithelium. Okay, so what do we have? Corneal epithelium conjunctival epithelium and lens, vesicle, or you can call it as a lens proper. So after understanding these diagrams, let us move towards the list of the structures from surface ectoderm. So we have a mnemonic for this surface ectoderm. It is L3EC2. Okay, we have it as L3EC2. So let us write first 3 times L and then we will see EC. So what are the structures from L? Those are the main lens, right? Then we get lids and glands of the eye. Then we have lacrimal gland, EG for lacrimal gland. Then we have the epithelium of cornea and conjunctiva. So epithelium of cornea and epithelium of conjunctiva. Okay. So these are all derived from surface ectoderm. Right? Now let us see the derivative of neuroectoderm. Okay. So after learning about the derivatives from surface ectoderm, we are left with this cup structure. This cup-like structure. And this is neuroectoderm. Now this cup-like structures develop further to form a cavity or cyst-like structure with 2 lips, inner lip and this one is outer lip. Both of this lip give rise to different structures. So to make it easy, I am drawing these lips into or from different colors. Okay, let me erase this. So let us draw the outer lip with black. Okay, here in between, outer lip with black. Okay, and the inner lip with green. Right. Now this outer lip continues to grow further and form the ciliary epithelium and iris epithelium. Okay, so down here as well, ciliary epithelium and iris epithelium. Right now it is just epithelium. The space inside these are hollow. Okay, these are just the epithelium. Okay, now this ciliary epithelium secretes the zonules. So what do we have? We have iris epithelium, ciliary epithelium and zonule. All these are from the outer lip. This outer lip also form the retinal pigment epithelium, and this inner lip give rise to the neurosensory retina, all the 9 layers. Between this inner lip and outer lip, we have a space. This space. This space is optic cavity. Okay. This optic cavity is a potential space which can lead to the further complication or is a most common site for retinal detachment to occur. So there is always, if you find a case of retinal detachment, it can be, or it's like most probably this may be because of the splitting between these two layers. Okay, so optic cavity. Now what else this inner lip give rise to? It give rise to the optic stalk or the optic nerve. Okay, so we have optic nerve, right? This inner lip also gave rise to vitreous gel. Vitreous gel, or you can call it as secondary vitreous. Right, it is given rise by inner lip. This outer lip also give rise to sphincter pupillae and the dilator pupillae. Okay, so we have over here sphincter pupillae and dilator pupillae. Okay, all these are the structures given rise by the neuroectoderm. So we will enumerate it first and then we'll go towards the mesoderm. So what are the structure given from neuroectoderm? First, the retinal pigment epithelium, neurosensory retina, all 9 layers. Then there is subretinal space, aka optic cavity. Then the fourth is optic nerve. Then we have ciliary epithelium, and sixth is iris epithelium. It also gives rise to the muscles present in iris. Those are sphincter pupillae and dilator pupillae. Then zonules, and the 9th one is vitreous gel. So all are these structures given rise by the neuroectoderm. With what? Neuroectoderm. Okay, now let us move towards the derivative of mesoderm. See, there is one rule which I think is like a good way to learn the derivatives of mesoderm, is that mesoderm always give rise to a connective tissue. What thing? The connective tissue. So first of all we'll move towards the, let me put the heading, mesoderm. So the first thing is vascular structure. So what are the vascular structures in the eye? The first thing when someone says the vascular structure in the eye comes, the first thing comes to your mind is most vascular layer or the part of the eye, that is choroid. So choroid, and second is hyaloid artery, which is also called as a primary vitreous, right? Then in muscles we get belly of extraocular muscles, right? And we also get third part, the temporal portion of sclera. So all these are the derivatives of mesoderm. Now what is the final topic we are left with, the derivatives, that is neural crest cells. So rather than learning the list, We'll first draw the structures of the eye which we got till now, and then we'll move towards the neural crest cell, because the remaining structures are derived from neural crest cells. Okay, you will come to know. Let's move towards the diagram of it. Okay, so I'm putting red color with surface ectoderm. So we have cornea, conjunctiva and the lid. Right. Now, and we also have the lens. I'm so sorry for the inconvenience. I'll put back things. So, red color for the surface ectoderm, and we have the cornea and the lid. Now, we have neuroectoderm. So, I'll put green color for the neuroectoderm. So, this is the optic stalk. Then we have the retina. Then we have ciliary body, and then we have the iris. So again, ciliary body and the iris. Okay, for surface ectoderm, I left to draw the lens, the main part. I'm really sorry for the inconvenience. This is lens. So for neuroectoderm, we got optic stalk, we got retina, we got ciliary epithelium, iris epithelium, and we got zonules. Then is sphincter pupillae and the dilator pupillae. I am drawing the iris thick because there are some remaining structures which need space to be drawn. So, okay, this is iris. We have vitreous. This is vitreous gel. Okay. Now for mesoderm, I am drawing in black. We have choroid. Since it is a vascular part, I am drawing it with like coil structure. Then we have bellies of extraocular muscle. That's it. What else we have is belly of extraocular, choroid and sclera. Okay, but we have only the temporal structure of sclera. Now, the remaining structures which I will be drawing in blue will be of neural crest cells. So let us first complete the cornea. So this is a cornea. Then we have conjunctiva. And then we have lids. Okay. So the first of all, neural crest cells give rise to the corneal endothelium. Okay. So this is corneal endothelium. This endothelium secretes Descemet's membrane. This is Descemet's membrane. Okay. Neural crest cell also give rise to the stroma and the anterior portion of Bowman's capsule. So let me draw the anterior portion of Bowman's capsule. This is Bowman's capsule. And this part is of stroma. Right. Now, we got all the structures like lid, conjunctiva and cornea ready. So let's move deep inside the eye, and here we reach to the iris. So this neural crest cells give rise to the iris stroma and ciliary stroma. So iris stroma and ciliary stroma. Okay. This neural crest cell also give rise to the ciliary muscles. So now we are done with the structure of iris and ciliary bodies, also zonules and lens. Okay, let us move towards the inside. So we got vitreous. Now this neural crest cell give rise to the remaining sclera. Okay, so we have remaining sclera. Okay, and also Tennant's capsule. So what is Tennant capsule? It is a fibrous layer which coats the eye. This neural crest cell also give rise to the tendons of extraocular muscles and also give rise to the oligodendrocyte, which secrete meningeal layer of the optic stalk or optic nerve. Okay, this neural crest cell, I'll put it in black, not black, in yellow, though it is a structure of neural crest cell, but since it is a colored or pigmented part of body, I am putting it in yellow, but it is a derivative of neural crest cell, and that is the melanocyte or melanin pigment in the iris. This neural crest cell also give rise to the orbital bones. So you got all these structures over here, that those are eye plus extraocular muscles and orbit plus glands. I forgot to mention the list of gland in surface ectoderm. So let us see here. So what all glands are there which are from surface ectoderm? So glands from surface ectoderm. The first main gland is lacrimal gland. Okay, this we agree with. Now we have M2Z. So what do you mean by the M2Z? These are all the glands of lid. So let me put the heading as glands of lid. So the first is gland of mole, second is gland of Zis, and from M we have, it should be S not Z, EIS, and the third we have Meibomian gland. All these are the glands of lid. Now we are left with the glands of conjunctiva or conjunctival glands. So glands of conjunctiva. So the first one we have is gland of Krause, then gland of Wolfing, goblet cells, gland of Henle, and gland of Mans. Alright, so here we are done with all the derivatives or origin of the tissue of the eye in the tissue in the eye.